Dom.
And so we had made it through my, excuse me? Whoop, it found me and lost me. All right, we'll start in the middle and maybe that'll give it what it needs. So we'd made it through the list of Don's gripes, and we'd gotten into what I think are the direction that things are going that specifically highlight what the shortcomings are and how now things should be changed. And so I was in the middle of telling you about my own lab's work. And so to back up just slightly, should we kill some of the lights in the room? Or, that when you look at single trial activity on even a small sample of neurons, and we can do this, we can do this analysis that I described on as few as half a dozen neurons, and we've gone up to 100 neurons, and it doesn't change the solutions at all. But we got basically, give me a sec. All right, got basically a time point when you have all of these neurons changing their firing, and we've explained how we analyze that, and so we get out of it. I don't really know why my computer is slowing down, but and part of that is you end up with the function that tells you how confident the analysis is that it's in a particular state, and that itself is something that that happens across a little bit of time, and that when you take a whole bunch of trials, the important thing that you find is that that transition happens at different times on different trials, and that explains why when you look across trials, you see what looks like a ramp, but really isn't. And so what we see is, in fact, something that, as far as we can tell, is instantaneous. We've done a lot of analysis on exactly how this occurs, taking into account the fact that you have to bin data in order to look at it, and there's some smoothing effects. And as far as we can tell, these transitions basically are instantaneous. And so going on one step more right now, one of the great things about studying taste, and another of the reasons I chose to study taste, is because if it is a decision, the outcome of this decision is, if this is a decision that the animal doesn't have to learn, the animal is going to make a decision about what to do with the taste in the mouth, it's also a decision that's very easy to observe. The animal, when it decides that it doesn't want something in its mouth, it makes a very obvious set of behaviors, most notable of which is called the gape, which is essentially this. And when the animal does that, it has decided to spit the taste out. And so we can do a very simple analysis in single trials of when we give a taste that the animal doesn't want, we can just simply compare the behavioral latency, the latency to it ejecting the fluid, to the latency of the time of that transition into the palatability-related state, or what we say is the palatability-related state. And so if all the data fall on this diagonal line, it means the two happen at the same time. And so if it falls on this side, it means the neural transition was first. If it falls on this side, it means the behavior was first. And it turns out that despite the huge variability, both in when that transition occurs and when the animal starts to behave, across all of these trials, you essentially get this beautiful function where the neural transition happens first, and immediately afterwards, the animal makes the behavior. And so this neural transition is, in fact, there predicting exactly when the animal is going to gape. We can look at a single trial and know not only that it's going to make this behavior, but when. And before we're done, I'm going to spend a little time a little later on talking about the experiment which shows us that, in fact, we know that it's driving the gape. We know that sensory cortex is driving the gape, but we'll get back to that. And this difference is 300 milliseconds. And so the nonlinear dynamics are the things you would expect from a system that doesn't work the way I described in the first lecture, from a system in which two regions are reciprocally connected, constantly interacting with one another and influencing one another, and that they are in fact making these nonlinearities occur via these feedback pathways. And so to put it simply, it should be the case that when you record from GC and whatever structure you think it is coupling with, you should see that these two regions are in fact doing this whole task together, rather than one doing its thing and the other doing something else, one doing its thing and then passing it on. And so we chose basolateral amygdala because it is reciprocally connected with GC, because it is known to be involved in taste processing, and it's known to be involved in emotion. And of course, liking or disliking a taste is an emotional response. And so my postdoc Abu Mahmoud has been doing those dual recordings, and what he sees is exactly what you would expect. First of all, I'm not presenting it here because this half of the talk, I wanted to make sure it fit into the schedule. BLA, basolateral amygdala, it turns out that it also goes through a sequence of state transitions while coding the taste. And meaning that it changed all of the neurons in BLA, suddenly changed their firing together. And that first transition, the first transition in cortex, there is a transition in BLA, but the two are not coupled. There is not above chance likelihood of these two transitions happening together in a coupled way, whereas the second transition, the transition into the palatability-related state, now these two are transitioning in a coupled fashion, which means that when the GC transitions earlier, BLA transitions earlier. When GC transitions later, BLA transitions later. But it's better than that, because it's not just correlated. It turns out that that transition happens essentially with zero lag. So the first transition, the ones that aren't coupled, actually BLA leads. This is ascending information into the system. It actually bifurcates in its pathway. Some of it goes to the basolateral amygdala. Another path goes thalamocortical. And it turns out that BLA goes into this taste response earlier in a way that is not coupled in time to when GC does it, or not significantly. And then by transition 2, they're not only coupled, they are functioning at zero lag. This is despite the fact that there are lengthy axons connecting them, despite the fact that there are one collection of cell bodies over here and one collection of cell bodies over here. In fact, they are essentially functioning as a single device. They are making these transitions together. Going forward, Abu did a whole bunch of analysis in a variety of ways. The only one I really want to share with you right now is he used a nice variant of Poisson general linear modeling to basically be able to estimate for every single gustatory cortical neuron that we recorded on the basis of, and I can tell you all about this if you want, but it really takes as model inputs other recorded neurons, stimulus timing, the history of the current neuron spike trains and other neuron spike trains. It has a cost function. And basically, it gives us a conservative estimate of what's coupled to what. Put another way, it allows us to estimate for each GC neuron whether it is involved in influencing BLA along the cortical amygdala pathway, whether it is being influenced by BLA along an amygdala cortical pathway, whether it is doing both, or whether it is doing neither. And when we do this analysis, we see that we have this nice Venn diagram of some neurons that are in fact influenced by BLA, some neurons that are influencing BLA, a whole bunch of them that are doing neither, and then a small subset that are doing both. And by the way, we did simple Bayesian analysis, and none of this is different from what you'd expect by chance, which is to say, given this number of neurons that are influenced by BLA, and given this number of neurons that are influencing BLA, you would exactly predict that number of neurons that are doing both just by chance. But when we do this, we can now do a division of function question that isn't by cell type, but is by where in the circuit it is. And so we asked the question for each of these GC neurons, each of these groups of GC neurons, how involved are they in taste processing by standard old-fashioned methods? How taste-specific are their responses? Given that I told you that these responses have multiple epochs, and the last one is palatability relatedness, how palatability related are their responses? And what we found was that for each of these, neurons that only output to the BLA, so these are neurons in GC, a lot of these neurons are neurons that take thalamocortical input and then send it to BLA. there is very little taste specificity in their responses. Meanwhile, the neurons that are actually taking input from BLA, that are influenced by BLA, are significantly taste sensitive, and they are distinctly more taste specific in their responses than the output neurons. And the same is true of palatability. In fact, I didn't plot it up here, but the neurons that are neither sending to BLA or receiving from BLA essentially are indistinguishable from the output neurons, whereas these guys are doing substantial amounts of both. Even more exciting, when we just take a look at the few neurons in the middle, the few neurons that are doing both sending of influence and receiving an influence, we see that they are the highest. And in fact, Abu takes the palatability relatedness of the response and the taste discriminability of the response. He combines those into a variable that he refers to as tastiness. And what he sees is that neurons that are both sending influence and receiving influence are even significantly more involved in taste processing than those that are just receiving influence. What does this mean? It means that what determines whether or not you are involved in taste processing is specifically how deeply in that reciprocal circuit you are embedded. If you are not embedded in the reciprocal circuit, you are not involved in taste processing. If you are partially embedded, you have some taste processing. The neurons that are most involved in taste processing aren't the ones that are receiving taste information from the periphery. They're the ones that are in that reciprocal network. So these findings are actually, it turns out, we were, of course, really, really excited and like, ooh, look what we've discovered. And then we went and looked at the literature and discovered that there are other people who are showing similar things in other systems. I don't have time to unpack these all for you today, but it turns out that there's multiple examples of systems in which a brief stimulus-driven transient in response is followed by a progression of coupled system activity through stimulus response states. Just so you have them, this includes Saravini et al. In 2019, who did intracranial EEG in humans from multiple visual cortical regions and found exactly this, that when they had subjects doing a picture naming task, when they would present the stimuli, they would see immediately or almost immediately responses that were basically stimulus-driven and not correlated. And then the system would basically be driven by that activity into a coupled state, and they would then see a coupled sequence of responses that reflected the processing of the stimulus. Similarly, Sup et al. In 2011 found almost exactly the same thing in surface EEG from multiple somatosensory regions in subjects that were simply responding with ow to a wrist shock. And so all of this is consistent with the idea of the separation of that separation of function, although not with the old, all of this is inconsistent with the idea of separation of function, although that doesn't mean it's inconsistent with the old data, it just means that the old data was interpreted in a way that was driven by the initial bias of the researchers, i.e., yes, when you look at these things, when you look for separation of function, you will find that regions aren't identical, but it turns out that that actually isn't because their function is separated, it's because the system isn't simply a ball of neurons. And so it also, by the way, is inconsistent with the easy interpretation of neural activity, and I should point out that all of this also says that, and including our data, that sensory regions are also doing motor processing. So this also knocks down the idea that you've got a progression of sensory to motor processing. Adding to the complication is some of my favorite work, which is on context dependence of function that comes with complex connectivity. It turns out that what follows from this and drives another nail into the coffin of the old computer modeling ideas is that it turns out that what neurons do depends upon the context. And this is actually known, but let me tell you about some really fun examples and how far we can take this. A little shout out to my fruit fly friends. So in Drosophila research right now, this is all the rage, and there are several examples that I can point to in which the function of an input depends upon where the animal is at the time. And keep in mind that we're talking about an animal that has only a few thousand neurons rather than the numbers that mice and rats and humans have. And so first from Leslie Griffith's lab, work showing that if you activate the detector of a chemical that is emitted both by food and by a mated female, activating this receptor, which you can do genetically and with optogenetics, it's attractive when the male is coming upon food, and that same input is repulsive when the animal is approaching a mated female. That the same activity of this neuron, this one neuron, can be involved in approach or avoidance depending on the context. Similarly, from Rebecca Wang, sugar, which the fly, by the way, senses through its feet, is appealing when hungry, and the opposite when the animal is interested in egg laying. So activating those inputs, the impact of that, what they are involved in, differs depending on the context. Finally, some of my favorite work from Gwyneth Card's lab. I mentioned earlier about looming stimuli. If you present a looming stimulus to a fly, you will activate looming input neurons, which causes the fly to duck and land if the animal is in flight at the time, and cause the animal to rise and take off if you present it when they're on the ground. And so these are situations when activation of the same neurons basically has different functions. But of course, one can say, yeah, but that's, it's connected to stuff in the middle, and maybe you're just talking about different shunting of the input. We can have even more fun than that. Even more troubling is context dependence in the impact of neural perturbations. And I should note that we recently published an opinions piece, and there are several examples of this that I'm not going to go into in the opinions piece. I'm just going to talk about the example from our lab, but this is also not uncommon. And so what I'm going to do now is I'm going to actually show you the natural progression of what I was talking about when I showed you that the GC transition into palatability comes just before the animal makes a behavioral decision. And so what we decided to do was simply block neural activity in GC during that process. And so this is just, finally, I decided to actually throw in a real anatomy slide. That's where GC is. That fluorescence shows you that these neurons have been infected with such that they express green fluorescent protein and also an optical silencer. And so then we can then light up that region and briefly silence those neurons. And when I say briefly, we had 4 trial types. One, which I'm not going to talk to you about at all, is one where we didn't. Well, actually, no. I'm not going to talk to you about one where we lit up GC for a long period of time. But in one set of trials, we actually activated this laser for half a second at the beginning of the trial, so right when the taste was delivered. Another group of trials in the same session in the same animal, we actually turned on the laser at 0.7 seconds. And another set of trials, we turned on the laser late. And so understand what's going on here. The range of transition times, of times that that neural transition happens from trial, which varies from trial to trial, and also the behavior can be like this distance. And so that means that pretty much this is always before it, this is almost always after it, but this half second of silencing is going to happen in the middle of that range of transition times. And what that means is that in some trials, we're going to block that transition, and in other trials, we're not. And so now looking at those data, what we see, and there's a lot of these data, and it's actually pretty complicated to explain, so I'm going to winnow it down to what's important. In the hands of Narendra Mukherjee, who is my graduate student who was doing this work, we have the neural perturbation comes on at this time. This is the average time of the behavior. So this is the average time at which the animal gapes to the nasty taste. And so that means that some range of times of gaping onset, again, with no laser, are actually this entire period with the average right there. And what that means is that there were some trials in which we turned the laser on before that transition had occurred. And we were recording at the same time, and so we could tell that in those trials, okay, no transition has happened yet. And while this is the average time of gaping without the laser, this is the average time of gaping with the laser. And so what we've done is we've massively delayed the onset of gaping. In fact, the animal doesn't gape until a couple hundred milliseconds after the laser turns off. For all intents and purposes, we keep the animal from responding by inactivating these neurons before this transition has occurred. But of course, the great part of this experiment is that quite naturally, there are also a small subset of trials where the earliest transitions occurred, and we didn't beat that. And so we have a small subset of trials where we can see in the neural data that this population transition already had occurred, and in those trials, we had no impact at all. Now this looks like we had an impact, looks like we made it earlier, but remember that this is just a small subset of trials in which the earliest transitions occurred and the earliest behaviors occurred. And so what this means is that essentially we had 0 impact. And that was true even if we missed that transition by 25, 50 milliseconds. Once that transition occurs, the animal has committed itself to the behavior, and nothing we can do can stop it. And so the point here for purposes of this talk is, boop. Hello? Okay, the point for this talk is that what this inactivation of these neurons do entirely depends upon exactly when we do it. And it's worse than that. It doesn't just depend on exactly when we do it. It isn't just the case that the function of these neurons changes over the course of a few milliseconds. It turns out that it has to do with what the system was up to before we actually turn on the laser. And so you can't, the question, what do these neurons do, not only depends upon exactly when you ask, but it depends on what the system was up to in the milliseconds preceding that. And beyond that, this question is ill-posed. There is no answer to the question, what do these neurons do? And so that brings me to the end of the talk. Things that sound good in neuroscience that aren't right. The question, what does that region do, is actually ill-posed. The function in which a group of neurons is involved will change from context to context and even from moment to moment. This shouldn't actually be surprising given that people are studying networks here. And that networks are in the brain, you never stop enlarging the network. And so there's the classic joke about how everyone's connected to Kevin Bacon by a few degrees if you just look close enough. Every neuron in the brain is in the context of every other neuron in the brain if you just expand your linkages a little bit. And in addition, the explanations that implicitly or explicitly characterize neural activity as either responding to some degree or not, that think that you can understand what a neuron is doing by answering the question, what does it respond to, are rather drastic oversimplifications. It's not just that neural responses are dynamic, it's that understanding that dynamic is almost certainly key to understanding function. And so it isn't, when we come back to something that I guess everyone in this room would agree with even before the talk, which is that we should be studying dynamics, I would say that not studying dynamics leads you astray in trying to understand what the brain is up to. And so these insights have extreme implications for how we think and talk about the brain. Given the constancy of interneuronal and interregional interactions, and given the fact that these constant interactions are doing the work of neuroprocessing, that the thing that completes the loop with the environment, that these interactions are what put us into the larger loop in the environment, my final, the most aggressively counterculture thing that I will suggest is that it might be reasonable to even ask what added value is achieved by constructs such as coding or representation. That one of the things that I recommend is that we just completely, what I think that all of this work shows is that we should just completely abandon the idea that something is coding something. Because when you talk about something coding something, you are implicitly suggesting that that does the work. That is involved in how things work. The reason that you see that line is because the V1 line detectors are active. And I think that what this work suggests is that we shouldn't be talking in those terms at all, that those terms are basically residuals from the computer metaphor, and that I would argue the brain doesn't code things. Or you want to say writ large, the entire brain might be coding something as a whole, but there is no subpart within that can be thought of as doing the coding or doing the coding of one thing or the coding of something else. And that is all I've got for you. A formalism that makes assumptions about how neurons and regions work together that neither make sense nor match the data. That's what I think of the idea of coding and representation, and that's it. I'd be happy to take any more questions.
Questions?
What should we use instead of representation and coding? Because you use the words yourself. You use the words yourself.
Yes, and obviously it's really kind of hard to avoid them. And it's funny because I went to graduate school in Indiana University, and maybe there are at least a few people in the room here who were as impacted by Hofstadter's book Gödel, Escher, Bach as I was. And so I actually got to work with Hofstadter, and I was working with him at a time when his big goal at that period of time was to rid his conversation of sexist references. And it was really hard. And not just because I knew him well, he was kind of a sexist. But in addition, just because it is so pervasive in our language, and that's even in English, which is not, you know, where the words aren't gendered. And so coding and representation are embedded in our language, and I would argue that that's why we use them. I would also, at another time, make an argument that one of the reasons that we need connections with philosophy is not because the philosophers are right in what they say, and not that we're wrong to suggest that philosophers should maybe run some experiments, but because one of the things that philosophy teaches you is to be very careful with your terminology, and we are not. We are sloppy, slovenly people with our terminology. The word is in our culture, we use it. I don't know what the answer is of what to replace it with. I don't know. But to be honest with you, I don't take that as my job. I don't accept the suggestion that you can't do science to show what's wrong unless you replace it with something else. That's not the way science works. Science works with disproof, not with proof. And so I don't know how to do it, and obviously it's hard to get rid of it yourself. Nonetheless, I think the key isn't that we get it right all the time. The key is that we should be constantly thinking about this. We should be bothering ourselves with this. We should never get comfortable with the language we're using.
So in physics, sometimes we describe the same system using various models which have various ranges of applicability. So I can say, okay, an atom is not an electron going around a nucleus, but that's good enough to calculate the spectrum of hydrogen atoms. And you have various other levels of models that you can use, and some more realistic models are far less useful in that sense because they're not give you exact answers. So even though, as we believe, this coding paradigm is not a correct description of the brain, to what extent do you think that's an effective model and that can be used for useful applications?
That's a great question. As always, as my friends will tell you, I'm here trying to elicit debate, and so I'm going as hard as I can on it. But clearly, these ideas have served a purpose. We've gotten where we are, and we've made progress using them. And I have no doubt that with more careful consideration, we can still make progress using them. But I would argue that what all this work shows is that it is time to move on to a new formalism. And this, you see this in physics all the time, that it's not that the work done in the past is null and void and should be abandoned. It's that once we've got special relativity, we realize, okay, we got to think about things a little differently in general. And so I think that, in my opinion, from here on forward, the only way that this metaphor and this terminology will continue to be useful is if we are at least much more aware of what we're doing as we do it. And if so, if people are willing, and I'm not talking with others, I mean with their own private spaces in their labs, etc., to say, okay, what am I really seeing here? Why am I using this terminology? I think that I don't think that it needs to be, maybe doesn't need to be 100% abandoned, but things get replaced.
Great talk. I'm not sure I really got like the presuppositions in the end, but if you cannot organize things like this first, this other things first, how could the brain process things? And like there's no like smallest information size that could be like a specific region. Everything's like is all over the brain. Like there's nothing that is like in a specific region.
So certainly. I think that the idea that everything is all over the brain is more broad than I'd like to go. I did, by the way, I did leave out, just because I thought I was going to run out of time, in Abu Mahmoud's new study where he was looking at the two regions together, he also did Granger spectral causality and showed something that is consistent with Antonio Damasio's work that I presented in the first half, that it does seem like there is influence heading from the BLA to the GC during the time when they're coupled that is asymmetric and could be thought of, when you talk about making use of ideas that are maybe a little obsolete, as basically sending the information about emotion to cortex for the cortex to act on. And so I don't think, I think that it is not necessary to go to just the whole brain does everything, but I do think that it's not clear that that any time-based dissection should be based on the ideas that we currently have. That it goes from that something's happening here, and then something's happening here, and then something's happening here. Within 50 milliseconds, activity is all over the place. And so, you know, we have a time-based division in our data, and we've seen it in other people's data as well, but it's a division that involves the functioning of the whole circuit. I don't know if that's a real answer to your question, but I think what I'm advocating is the leaving behind of the idea that the breakdown works sensory to motor, that you do sensory processing, and then you get to intersensory integration processing, and then you get to motor processing, and that we stop saying it, so that it's these brain areas and then these brain areas. I think that that stuff just isn't the way the brain works.
Oh, thank you very much for the talk. Oh, it's here. I'd like to ask about methods, because you brought up the idea of using nonlinear methods to analyze, and I think that's very important. That's what brings interesting phenomena to the brain. And although we have very robust linear techniques for statistics, our mathematical tools for dealing with nonlinearity, especially dealing with directionality, causality, are very
sucky.
Yeah, exactly. Where do you think we are in terms of the tools we need to get these new insights?
Yep. I think progress is being made all the time. And having said that, I'll admit that I am a consumer and not a producer. And so that also means that I'm not really an expert. But I will say, and this is maybe the most optimistic thing I'll say today, is that I don't think that we have to get to great techniques tomorrow. I think that one of the things that, I mean, we were talking about this at lunch. I'm driven in science by just the sheer joy of the intellectual pursuit, and I don't care where my papers go or anything like that. And so I will continue to use the techniques that are available, recognizing their shortcomings and trying to, you know, make allowances. You know, one great example for me is that we're using hidden Markov modeling, which is a really fun technique, but that makes all sorts of assumptions that are ridiculous. One of them is that, okay, you find these sudden transitions, and the assumption is that everything that's happening before the transition and after the transition is purely stochastic and stationary. And of course that's not true. And so now what we're doing is we're trying to use the next generation of techniques to look within states and see if we can see the intrastate dynamics that are ignored by these older techniques. And you know, those techniques that we're using, they have all sorts of horrible shortcomings. And we're going to make like a little step. And we're going to, if you talk to me next year, in fact, my graduate student just sent me the data. But it's still going to be sucky. But it's going to be a little less sucky. And so I guess I'm not so pessimistic, despite the fact that you are absolutely correct, that we are currently not very far along in developing those sorts of techniques. And I think part of the reason is we don't have enough of the field mobilized in that direction. But I think the best tool we have, he says, knowing that this is going to sound really hokey and foofy, is the human mind. And you can take whatever you got and make whatever you can of it. The important thing is that you are asking the right kinds of questions.
If you want to establish like a quota system for question asking, I understand because I have so many of those. But so when you talk about, say, a computational paradigm, I think, like you said, you have to be very precise about language because in a way, you could define computation in a way that what the brain does is computation by definition. So I'm trying to think of what the elements of what we usually think of computation are, which elements they are that you are actually questioning. One thing that comes to me is the idea of a sort of feed-forward paradigm where you have one modulus that sends information to another one, that sends information to another one, always in one direction.
Right.
And another thing that occurs to me often in brain things is that the architecture in small scales and large scales tend to be similar. So we're looking for some sort of general paradigm of how information is transmitted. And I'd like to propose that one way you can think of this post-computational paradigm is to think that you never have like one direction flow of information, but rather, and that's true for attention, for single neurons, for whatever, you have always a feedback loop that essentially filters and selects what gets passed on in terms of expectations and outcomes. So I was wondering if you have any thoughts on that as a potential new pre-paradigm for what computation looks like in the future.
Right. So I have to tell you, you just asked a lot of questions. And I'm completely down with that. In fact, when this is all done, you should feel free to email me, and I'll be back in Boston, but I'll be happy to continue this conversation as long as you want. And so a couple of things, and I'm pretty sure I'm not going to cover the waterfront in what you said, but with regard to computation, yes, I recommend for everyone an article that was written, I think, in the journal of I think it was written in the Journal of Philosophy by a guy named Van Gelder, way back in the 1980s or 90s. We were talking about how far back some of this goes, and the article is called "What Might Cognition Be If Not Computation?" And it makes the distinction that you just made. Now, he misidentifies it as saying he's opposed to computation, when, as you point out, the stuff that we're talking about, the brain does computation. What he's really arguing against is this idea of what he would probably now call an information processing approach, where something happens and then something else happens and then something else happens, in favor of a dynamical approach. And he does this lovely analogy. He uses as a metaphor a device called the centrifugal governor, which was one of the early devices made to cut logs when people needed tons of houses. And it worked on dynamical principles, and it did computation. It just didn't have a sensor for this that then sent that output to that and stuff like that. And so I would say that, yes, what this stuff advocates against is essentially the parcelization of function into different. And so then, with regard to the last part of what you're saying, and I think we're probably going to have to, I probably need to ask you some questions to get deeper into this. That one of the things that's sort of hidden in what you're saying is that, of course, once you go down this road, then all sorts of stuff that's happening before the stimulus even arrives is a part of stimulus processing. In taste, one of my favorite examples of this is if you've ever had the experience of coming up to a table and seeing an amber beverage, and you say, ah, ginger ale, love it. Yes. And you put it in your mouth, and it turns out that it was apple juice. Regardless, you can like apple juice and like ginger ale, and the experience is not either of those. In fact, it is reliably aversive, and this is because what these inputs that you're activating with this drink do entirely depends on what you were expecting. And so we are very interested in that. We've got a lot of stuff going on in that. But with rats, it's a lot harder than with humans. And so I'd be delighted to talk to you about that kind of stuff offline.
So Don, actually I have a some provocative question. Right? So, what's the effect, for example, a neuron? Right? It's a totally wrong thing to look at to think about the brain. Because I'm thinking, for example, right, if you think about muscles, I think it's a good example. If you want to understand the function of a muscle, probably just studying the muscle cells, you know, it's almost useless. Or you will find some correlation, right? Probably the muscle that's in your arm will be related to the arm movement, and probably the muscles that are closer to the junction probably is more related to the rotation, whatever. So you couldn't associate function, but if you really want to understand a function of the muscle, you really has to look at the entire muscle scheme to understand what's going on. So it could be that, I'm just thinking that the brain is not so far away maybe from the muscle, and maybe it could be a similar kind of situation. What do you think?
Yes, so I'm afraid that I can't take the bait because I agree with everything you said. I think that the way we look at the brain, we do take the single neuron activity, but we process the hell out of it because we think what's important are these network state changes, and we think that even the neurons that we're recording are just giving us a window into the fact that this process is occurring, but probably they are also not the things that are doing the work. And then we saw the talk, I mean, I missed the first part of it, but the talk we heard this morning, the idea that it has to do with criticality, that of course itself is something we could have a debate on, but if it's really about whether the network is near criticality, it doesn't really matter what each individual neuron is doing in that context. I think that these are great questions, and I think that everything that we think is right now will eventually be replaced by something else, and I look forward to that. So right now I'm recording from neurons because it's the best thing that I know to do, and it's giving me this window in, but I can see a time in which we stop worrying about neurons altogether and start worrying about some abstract idea of the network state.
Any more question? Okay, let's thank Don again.
Thank you all.